Today is Monday, April 24th, 2017. My name is Meena Rao, and I'm at the McCormick Convention Center in Chicago attending EB 2017. This afternoon, I have the distinct pleasure and honor of interviewing a dear friend, a colleague, Dr. Primal de Lanerolle, for the American Physiological Society's Living History Project. Primal has been a member of the APS since 1985. He's a professor of physiology and biophysics at the University of Illinois at Chicago. He joined UIC in 1984, and where he has honed his exciting and independent research career. As a cell physiologist, he has contributed much to our understanding of cellular movement. Primal has published over 100 papers and is sought after as a peer reviewer and as an invited speaker. His work sits at that sweet spot in biology where both NIH and NSF consider his projects as worthy of funding, and he's received many awards. Primal, welcome to the Living History Project, and thank you for agreeing to be interviewed for this series.
Well, thank you, Meena, for the very nice introduction.
So, Primal, first let's share with our listeners some information about your early life and how those influences your parents, your family, friends, teachers, books that contributed to your career development.
So, I was born in Colombo, Sri Lanka, and I grew up there, and my family moved to the United States in 1958. We first moved to Southern California, and then from Southern California we moved up to Berkeley, where I grew up. My biggest influence in my life was clearly my parents. My father was educated in the United States. He came to the U.S. On a scholarship from the YMCA and went to Springfield College. He was very interested in the arts and also the sciences, and he pushed both my brother and I to become physicians. And he was the biggest influence, and he was the person who really motivated us towards academics.
So you were very young when your family migrated from Sri Lanka, and yet you were old enough to remember your transit. You arrived at an impressionable age, around 12, I think, and I believe you had to plunge into middle school, which is really one of the most challenging periods of a kid's educational, in a strange land. So how did those first impressions as an expat and immigrant shape your thinking?
Well, moving to the U.S. was a very difficult experience. One has to remember that we moved in 1958, and we got on a ship, and we took a ship from Colombo to London, and took another ship from London to New York. And when we got to New York, arrived in New York on July 3rd, 1958. And the next day, as we were walking around, there was a big parade, and we thought, God, what a great country. They're having a parade because we got here. And obviously not the case. From New York, we spent about 10 days in New York, and from New York, we took a Greyhound bus all the way across the country to Southern California, where we stayed with a friend, Ben Gautier. Ben was a soldier in the U.S. Army in India during World War II, and he used to come to Sri Lanka and visit the YMCA on R&R, and that was where my father worked. And he made friends with my parents, and he eventually ended up sponsoring us and our move to the United States. Ben in time became a really great friend. I was his best man at his 4th wedding, and this was after a quadruple bypass and 2 knee replacements. He was really a guy. And so we stayed with Ben, and eventually my father got a job in Berkeley, and we moved to Berkeley. Berkeley has always had the reputation of being very liberal, and perhaps Berkeley was very liberal at that time as well, compared to the rest of the country. But growing up there was truly a mixed experience. On one hand, I used to love going up to the Cal campus. I used to cut classes when I was in high school, and I would go up to the Cal library, and in the 50s, you could you could go anywhere. You could walk around the stacks, and I used to spend the afternoons just looking through the stacks and finding old books that I could thumb through and read. And Berkeley really instilled in me an attitude and an appreciation of academics, and made me want to go into academics. On the other hand, Berkeley was also very racist. It's hard to believe, but and it kind of takes me aback sometimes when I think about it. But we were the first Sri Lankan family to emigrate to California. There are over 100,000 Sri Lankans in just Southern California alone. But we were the first. And remember, there was no email, there was no internet, there was no, you wrote a letter on an airmail form and you put it in the mail, and it took 2 weeks to get back to Sri Lanka. So we were basically cut off from all of our friends and acquaintances. It was also difficult because we couldn't buy a house where we wanted to. There were a lot of racist aspects to Berkeley. And when my mother would walk down the street wearing a sari, people would stop and stare because they didn't know what a sari was. Hard to imagine nowadays, but they didn't know what a sari was. Perhaps the most difficult part of growing up in Berkeley was that my junior high school counselor decided that I wasn't smart enough to go to college. And so she had me pegged for the remedial classes and trade classes, and I had to fight to get into college prep classes. She even told my father that I was too dull and that I should only go to a trade class. And that was very hard, and having to deal with that was not easy. But eventually I did, and I have to admit that when I got my PhD, I was really tempted to write to my junior high school counselor and thank her for the motivation. But it was a difficult time, and it was a mixed bag. It wasn't, in some ways it was very difficult, in other ways it really stimulated me to go into academics. Fortunately, there were a couple of teachers at Berkeley High who took an interest in me and started challenging me. And I think that's the biggest thing is because I was not white, and because there were no Indian students in the Berkeley school system. I mean, it's hard, it's inconceivable now, but there were no Indian students. We were the first, my brother and I were the first two Indian students or from the Indian subcontinent in that school system. They didn't know how to deal with us, and they didn't know how to challenge us, and didn't know how to basically didn't think we were worthy of the effort.
Oh. So Primal, you've often spoken about, so from Berkeley High, you've spoken about the power of a liberal arts education. So from Berkeley High, you went to Whitworth College. And can you speak briefly about how Whitworth helped influence you and shape you as a scientist?
Whitworth was a revelation. Whitworth was fantastic. It's a small liberal arts Presbyterian school in Spokane, Washington, and what was great about Whitworth is they were willing to tolerate, and I think in somewhat of the Presbyterian spirit, to accommodate differences of opinion. And I wasn't the typical student there, but Whitworth really nurtured me, and Whitworth really encouraged me. I had gone to Whitworth, as I said. My father had been very committed to higher education, and he had really pushed both my brother and I to go to medical school. And I went to Whitworth with the idea of going through a pre-med curriculum and going to medical school. But I fell in love with the humanities. I fell in love with my literature classes, mostly because the literature and history professors that I had were absolutely fabulous. They were absolutely fabulous, and they really stimulated me to think and to challenge myself. So eventually, I ended up getting a double major in English and chemistry. Interestingly, even though and I really focused on, I really enjoyed my English classes, but I still took a fair number of science classes, and even though I didn't do particularly well academically in them, I must have learned something. In particular, I took a quantitative analysis class that, boy, I encourage every student to take some quantitative analysis, because you learn the importance of being precise. You know, being precise. Science is about being precise. So eventually, but the thing about literature is that literature, when you do it in a really analytical sort of a way, you still have to pay attention to detail. You know, in my first, as a freshman in college, I took an American Lit class, and one of the questions had to do with one word in a very long story, and I didn't get it. I missed it. You know, and after I read that question, I got my exam back. I thought to myself, you better start reading a little bit more carefully, and that's something that's carried me through the sciences as well.
Fantastic. So your post-baccalaureate years were very interesting in political times, right? It was the late 60s, and there again, that time period in history was pivotal in your career choice. Could you just spend a couple of minutes talking about that, because I want to spend more time about then your research path and what you're actually doing in research right now.
Okay, I'll try to be short. Mm-hmm. So when I graduated from college in 1968, and 1968 was 1968, but it was a pivotal year in our history. The Vietnam War was really ratcheting up, and I was at San Francisco State, where they had the black student strike and the Cambodia bombings. I mean, the country was like in flames. It was like the country was going to go down the tubes. And while I was there, I had. The summer previously, I had worked at a hospital doing some research, some medical research. And while I was a graduate student working on my master's in literature, the Army wrote to me and said, "We have plans for you. We're getting ready to send you to Vietnam." To make a long story short, with the help of a lot of doctors at this hospital, I applied for something called alternate service, which is where you could spend 2 years in service to the country in lieu of going in the Army. And I appeared at my draft board, and they said, okay, and I did that for 2 years, which fulfilled my military obligation. But that period of time was really a turning point in my life, because at the same time I was working on my master's, and I was understanding that I really liked science. And what I liked about science is that it was the creativity of science. It was the fact that I felt that the type of creativity I had applied better to designing experiments and to doing experiments and to proposing hypotheses, formulating hypotheses, than it played to writing or being an author. So in the end, after I did 2 years of service, we got married, went to Europe, came back, finished my master's, and I went to graduate school.
Great. Well, thank you for that, Primal. So talk a little now about graduate school, mentorship. From graduate school, you were at UC San Diego, and then you decided to traipse across the country to NIH for a postdoctoral fellowship. And I think especially for our junior listeners, it would be nice to find out what were your challenges, what were the fantastic things you learned in graduate school, and how that helped you find your postdoctoral position. I'm going to give you about 5 minutes for that.
Okay. So I went to graduate school at UCSD, and why I got into UCSD is still not clear to me.
It's okay.
And but there were two people there, John West and Palmer Taylor. And when I interviewed there, I think they were taken by me. And I think they were taken by the fact that this crazy person wanted to do a PhD in physiology and pharmacology after just having completed this, a thesis in English literature and written a thesis on Milton. And so they accepted me, and of course I was totally unprepared for graduate school. And it was a very, very difficult time. And I have to admit that I almost flunked out of graduate school. But at a point, I had to decide, I made the conscious decision that if I was not going to succeed, it was not going to be because I hadn't tried my best. If I wasn't going to succeed, it was because I had failed, not because I didn't want to go home whining that I hadn't tried hard enough. And I think that has been a really important, and of course I worked hard and I succeeded. But that has been a really important principle in my life. The best way to guarantee failure is not to try. So it's really important to try your best. In graduate school, John West both John and Palmer are fantastic people, terrific minds, and they have really broad educations and experience. So they were always fun to talk to and always stimulating to be around. And Dr. West supported me for almost the entire period I was a graduate student of he had a pulmonary training grant. I ended up working in Jim Stahl's lab, and Jim was a new assistant professor who had come there, and I was Jim's first student. And as a young assistant professor, he had 4 people in his lab, including me, 3 of whom went on to be professors at very good universities, and that's the kind of students you want to start off with. Jim taught me a lot. Jim was a very rigorous scientist, who, like my professors in English, taught me the importance of paying attention to detail, and because the devil is in the detail. And Jim also went further by emphasizing the importance of being quantitative and really knowing that what you're doing is in fact can be substantiated on some quantitative basis. So those were really important lessons, and those are lessons that I try to communicate to other students. From NIH, from UCSD, we moved to NIH. I worked for Bob Adelstein at NIH. Bob had figured out the myosin phosphorylation mechanism, which is such a cornerstone in cell biology and physiology and signaling. I mean, it regulates so many important processes. And Bob was also very, very quantitative. But Bob also emphasized, Bob was very intuitive. He followed his nose about what he thought was interesting or may result in some really important observations. And he taught me the importance, or he emphasized the importance of having a good nose in science, of being able to sniff out which of these 5 issues or these observations that you have is going to be the most productive, is going to be the most interesting. So so Bob was great that way. It was a very productive time for me, and I enjoyed it greatly. There's one other thing that I want to tell you about, and one of the things that I did is somehow or another, through a sort of a long process, I ended up starting a softball team, because there was a softball league at NIH. And I ended up starting this softball team. And the first year we were terrible. The next year we were a little bit less terrible. But by the 4th year, we were a great team. And that experience taught me so much about relating to people, about fitting people into what they were best at, and asking only what they could really, challenging them, pushing them to do better, but not over-expecting from them. And so that is it's sort of a funny thing to say, but playing softball at NIH was also really instrumental in my career.
It also taught you how to play hardball in science, huh? So your research program, Primal, has evolved from myosin phosphorylation and smooth muscle contraction to non-muscle cell motility. And not everyone would perhaps view this as a natural move, because there's still so much to understand about smooth muscle and smooth muscle function. So why digress? Why the shift? What made you do that? And can you tell us a little bit more about your work with nuclear myosin and the controversies surrounding it?
Okay. So I started off in Jim's lab working on smooth muscle, and I worked on tracheal muscle because I was supported of this respiratory grant, and I knew something about respiratory, and I had an interest in respiratory. And
John West was your professor.
And John West was my professor. My project as a graduate student was to figure out whether myosin got phosphorylated when smooth muscle contracted, and I was able to do that very successfully. When I got done with my PhD and I was looking for postdoctoral opportunities, I wrote to some people who worked on smooth muscle, but I felt that science in smooth muscle had progressed to a point, and I could see that it was a little bit blocked. The interesting things, there were a lot of interesting things, but they were going to take some experimental advances to really to address. The other thing is that I knew, or I had the intuition, or I had the nose that my nose told me that myosin phosphorylation was going to be really important in things like cell motility and cell division, which was then going to play really important roles in cancer and in development and in embryogenesis, and these are big parts of cell biology, and so I felt that it would be good to learn some cell biology, and Bob, and that's why I went to Bob's lab, because Bob had studied platelets and had studied non-muscle cells and myosin phosphorylation in non-muscle cells. So I really made the shift to studying smooth muscle, to studying non-muscle cells, because I felt that there were more possibilities and there were more implications, at least in the short run, with non-muscle cells than smooth muscle cells.
Great. So now you want can I segue into the next question, which is so when you first published your work, fantastic work on nuclear myosin, it was controversial. And the excitement was not without its challenges, obviously. So how did you deal with that, and what were your take-home lessons from that? You're a fighter, so...
Yeah, well, don't give up. That's the first thing you can't do. So the myosin 1 story is very interesting. And I need to give a little history to put it into perspective. I had a postdoc, an MD PhD, who had come from Poland, Grzegorz Nowak, a very talented, smart guy. And he started doing some things, and I had, and what I like to do with postdocs is to give them some subject areas and some reading, and so just go do some reading and kind of figure out, fill in the background, figure out what you really want to do, and then let's talk and plan a project. And one of the things that I did was to give Grzegorz a paper that had been published by Joe Albanese, a JCell biology paper, in which he had described making purifying myosin 1, and the first unconventional myosin that was described by Tom Pollard and Ed Korn. And And so I suggested to Gregors that he take this paper and think about purifying myosin 1 and making an antibody to it. Because I thought that if he had an antibody, I had made an antibody in Bob Adelstein's lab to myosin light chain kinase, and it had been incredibly useful. So I thought an antibody to myosin 1 would also be useful. So Gregors goes out, he purifies some myosin 1. I show him how to inoculate rabbits and how to bleed them. And he starts bleeding the rabbits. And one day he comes into my lab with this really long face. I mean, he looks so sad and so unhappy. And I looked up and said, what's the matter? And he looks at me and he says, well, Primal, you know, that antibody that we made, there's something wrong with it. I said, what? And he said, well, it stains the nucleus. And I said, well, do you have confidence in your experiment? I mean, did you set up the experiment right? You affinity purify this? Do you think the secondary antibody doesn't stain the nucleus? I mean, no, I think the experiment is fine, but it's staining the nucleus. And I said, well, what's wrong with that? And he tells me, well, everybody knows that there's no myosin in the nucleus. So I laughed and I said, let me take a look at it. And then all the nuclei were staining. And I told him, well, you know, you either discovered something really important or you've caused me a lot of pain, but we better find out. One of the things that I tell my students is you can't ignore data because the dogma tells you it's some other way or you don't like it. You know, there's no such thing as I don't like this data. It's a question of did you do the experiment properly or not? If you did the experiment properly, you had to relate to your result. And in many cases, the best results are the ones that are totally unexpected, that tell you something that you didn't expect. So to make a long, you know, eventually we were able to show that that there was a protein that looked like myosin. Eventually we cloned the gene, and we were able to publish the data. And this is again another lesson in never giving up. When I first submitted grants on myosin 1, they went to cell biology study sections. And the comments that I got were really interesting. One person told me that one reviewer told me that one of the best established dogmas in cell biology was that there was no myosin in the nucleus. And you know, I said, this is a stupid grant and triage. Somebody else told me that it was not worth funding me because I was a physiologist. This is absolutely true. The comment was that I was a physiologist and I wouldn't even know what to do with the gene if I cloned it. And well, we published a paper in Science and I got the grant funded. But I got the grant funded because of the goodness of the program officers at NIGMS.
Neat story of encouraging students to go by their data and not by what the dogma is. So you've always tried to maintain your research at the cutting edge, and not for the sake, and as we've just found out, not for the sake of a technique, but for the purpose of understanding a fundamental physiological question. You've succeeded in making your science attractive both to NIH and NSF, which is very unusual. And can you tell our listeners a little bit about how you managed to sit at that sweet spot and optimize your interactions with these two funding agencies? I'm not really sure, frankly. Luck of the draw, huh?
Well, I think there's no doubt that things have changed in the last 20 years. And right now, methods and techniques are very much being emphasized by funding agencies. I think part of the reason I've had success is it was a little bit different time and place, where people understood that what's really important, and I tell my students all the time, the thing that will distinguish them as scientists is the quality of the questions that they ask. And I think this is sort of a long-winded way of saying that ultimately where I profited was that people understood that I was asking important questions, big questions, tough questions, not easy questions to answer.
Great. So, Primal, I'm cognizant of time, and I want you to spend just a minute or two telling us about just what you feel are your most seminal findings, and what has excited you the most, and how you've taken that in a new direction. And in some ways, we've covered part of it, but if you can just summarize that, then I want you to speak briefly about your mentoring philosophy, which you again have touched upon. And towards the end, I want to ask you a few other questions. So we are cognizant of time. We've got 14 minutes more.
So we, without doubt, the discovery of myosin 1 has been really, really important. In addition to myosin 1, we also published a series of papers on the role of actin in the nucleus. Actin has, we've known about that there's actin in the nucleus for over 50 years or 40 years, but what it was doing in the nucleus was not really clear, and we showed that it was absolutely essential for forming pre-initiation complexes and the activation of RNA polymerase II. And we also published a paper showing that it was in collaboration with Ingrid Grump's group, showing that it was important in activating POL1. I think those two papers, along with some papers with a paper by Jerry Crabtree and Maria Vatian, and another paper by Maria Vatian and Richard Treisman, were really the seminal papers in jumpstarting interest in nuclear structure and how the nucleus is organized and how it might be compartmentalized into domains. That's clearly important. I think our work on myosin kinase and myosin phosphorylation is also important, especially the connection to GTPase pathways and the regulation of myosin kinase activity by ERK and PAC. So I think both are equally important.
Fantastic. And we look forward to many more new discoveries and directions that you're going to set. So can you tell us a little bit about what you've mentored a number of students, postdoc fellows who are all successful in their careers, have set up independent careers. What were your goalposts for mentoring, and what advice can you give students to look for, in terms of what they could look for in a good mentor?
Yeah, so I like to say that picking an advisor and picking a spouse are the two decisions that get the least, far too little thought than they deserve, because you're going to live with somebody for 4 or 5 years. And I think one of the most important things is to respect your mentor, both as a scientist and as a person. Times inevitably will get rocky. And if you don't respect each other as people, it's going to be really tough. With my students, the things that I've tried to emphasize to them is that I want them to push themselves. It's very easy to become complacent, to be comfortable in what you do and not try to extend beyond yourself, and that's not a good approach. You really need to push yourself, and to a great extent, I can push students, but that motivation really has to come from inside you. You have to be passionate about science, or you have to be passionate about anything if you really want to be good at it. Eventually to be really good at anything, you're going to be competing internationally. And to do that, you have to take pleasure in the process. So I try to get students to understand who they are, to understand what gives them pleasure. If you don't like doing experiments, then do something else. Yeah. So I try to get people to understand what motivates them, what gives them pleasure, and then to push themselves. One of the things that I try hard to do with my students is I remind them that if they are probably only going to write one PhD thesis in their life. And I'm very proud of my thesis. And when my wife worked at Catholic University, and by happenstance, she got my thesis bound in leather by the bookbinder to the Pope at that time. And so one of the things that I do now is I show students my thesis and I tell them, I want you to have something like this that you can be proud of for the rest of your life. And I have all my students give me 2 copies of their thesis. And I get that bound in leather. There's a bookbinder near here. And I get it bound in leather and I give them 1 copy and I keep 1 copy. I try to emphasize this is something, I want you to be proud of this for the rest of your life.
Mm-hmm. Oh, what a wonderful sentiment. I didn't know that. So, Primal, in your view, what contributed to your success both at UIC and in your research career?
Well, well, you have to be a little lucky. Yeah, I mean, you have to be lucky. But the harder you work, and the more passionate you are about what you do, the more likely you're going to be lucky. Yeah? And I think so. So I think I have been lucky, but I also feel, it's like John Lennon said, 75% of success is showing up, and that's true. If you show up and you keep plugging away, it eventually works. I always tell my students, and do good science. Do good science. Do hard, well-disciplined, rigorous science. Be as demanding of yourself as you are of other people. It's really easy to criticize somebody else's paper, but be as critical about yourself. And I think it's a combination, partly of luck, partly of a lot of hard work, but a lot of it is also because I enjoyed the process. I enjoyed working in the lab. I enjoyed being with my students. And I think it's those.
Great. So, Primal, in the last few minutes, I would be very remiss if I did not ask you to tell us a little bit about Sandy, your wife of nearly 45 plus?
48.
48 years, and your lovely daughter, Rena. So what roles have they played in your career and life?
Well, what can you say? So we've been married almost 48 years, and really, I really can't take a whole lot of credit for that. I mean, Sandy's an incredibly patient person, and that's clearly, that's clearly the key to the success of our marriage. I mean, Sandy has been very supportive. I've taken her from San Francisco to San Diego to Washington D.C. to Chicago, and she has been very good spirit, and I couldn't have done it without her. There's just no 2 ways. Rena has been a joy, and I think the biggest thing that Rena has taught me is patience. Is having patience with students and other people.
So, Primal, our conversation so far makes you sound like this lab-going nerd, but many of us know that you're really a multifaceted person. So, in addition to this little softball vignette, you have a number of avocational interests: sports, literature, literary pursuits. A sentence or 2 about how that has helped you shape or maintain your focus in science as well, how that has complemented your scientific activities, and then leave time for one final question.
Okay. I've played a lot of sports. I used to play a lot of basketball, which you would think was not an intuitively obvious thing for me to do. But I like baseball a lot, and I've played racquetball until very recently, and I still work out quite a bit. Working out and being in shape has been a surprisingly important part of my academic life. I think it has led to my stamina, to my ability to work long hours. It's also been a fantastic stress relaxer. And there's nothing like hitting your partner with a racquetball to relieve stress.
Of hooking that fish.
Oh, and then the other thing is I like to fly fish, and fly fishing has been a great way to relax and to let off steam, to let off pressure.
So, Primal, in your CV, one of your coolest recent projects is something called Ralame Rides Again. You want to tell us about that?
Yeah, Ralahami Rides Again. Ralahami Rides Again is a book of plays that was written by my father. My father was an editorial columnist in the two largest papers in Sri Lanka, and he was a political satirist. And he started writing these plays in 1927 when he was at the YMCA. And they were plays that bitterly criticized, in most cases, first the British government, made a lot of fun of the British, and then later on the Sri Lankan government. He was very interesting because one of the basic tenets of these plays, he argued against the Sri Lankan government changing the official language from English back to Sinhalese. Because in the 40s and 50s, he argued that if you change it back, you're going to disenfranchise a significant minority of Tamil people who have been in Sri Lanka for decades. And he said, and the other thing he also said is that we're a country of 10 million people. Nobody else speaks Sinhalese. Nobody else cares about Sinhalese. We need to speak English so that our kids can compete in a global market. This was long before anybody else was. I mean, he understood how divisive language is. If you have a different religion, you can go to your synagogue, you can go to your temple, you can practice your religion at home. But if you don't speak the language, you can't have a government job, you can't get an education. You can't be part of the legal system. And these were prescient observations that he had. And so he had done this series of plays starting in 1927. The last play was in 1957, just before we left Sri Lanka. And I compiled all of his plays into a single book, and I edited them. And I wrote an introduction and edited an introduction that he had written. And we released it in Sri Lanka about 3 years ago. And I'm just really thrilled because about 5 of his plays now have been done since I released them. And the plays seem to be still relevant. And the issues, one of his plays is called 50-50 because to guarantee equal civil rights, his solution to the race problems in Sri Lanka was to intermarry. Because if you intermarry, then the kids are going to be 50-50. And you don't have to worry about representation at that point. So he was quite a character. I mean, and this was something I was very pleased to do for him. The other thing is I got to talk about my mom. I need a couple minutes. I got to talk about my mom. My mom, was the glue in our family, without doubt, and I was very close to my mom, and she had started a Montessori school in 1975 in the Bay Area. And she passed away in 1994, and and before she passed away, she asked my wife and I to continue running the school, and we've run that school since 1994, so for over almost 25 years now. And we are approaching our 50th anniversary. It was something, being Asian, making your parents proud is important, and I'm happy that I was able to do this. My father wanted me to go to medical school, and even after I was a professor, when I was a postdoc in my 30s at NIH, he said, you know, you can still go to medical school. But eventually he was very, very proud of the fact that I was a professor at UIC. So it's...
Comes around in a full circle.
Yeah, it's terrific to be able to.
So the last 1 minute, Primal, is can you describe the role APS has played in your academic life, and very briefly, what advice would you have for new researchers and physiologists in the 21st century?
The APS has been really important because I think the APS, especially when I was getting started as a young assistant professor, the APS was very supportive. Marty was very supportive, and I think made me feel more accepted as a physiologist and as part of the physiology community. For young people, my advice is be passionate about what you do. You know, it's really important to know who you are. It's really important to know what makes you happy, and find something you're really passionate about and make that your career. And the one thing that I do want to emphasize is that, you know, given my background in English literature, I've always wanted to be a scholar, you know, and a scholar is being a good scientist, but being good at your discipline, but also being a good teacher and being a good mentor and being and discovering new things. And in literature, and you know, as scholars, we are looking for truth. And I think we think of that as a process that we leave to people who are in the humanities or in religion, you know, in literature or religion. But we as scientists are also looking for truth. And the fantastic thing is that we have a little bit more objective way of deciding what is true or what is not true. And that process of looking for truth is really almost a divine responsibility. You know, it's something very special that not many people get to do. And my advice is treat it with the proper respect.
Great. Thank you so much, Primal. This was a really fun and informative interview for APS's Living History Project. It's been a real honor for APS to learn about your life and career, and for me, actually. Thank you so much on behalf of the Society.
No, thank you, Meena.